So, another part of evolution, diversity and systematic, we start first three domains system. So, three domain system, the universal common ancestor and the tree of life. Okay, universal common ancestor is abbreviated as LUCA, Luca. That means last universal common ancestor. Okay, another term is there, one which is known as FUCA, Luca, first universal common ancestor. That is the life form after the prime, primitive cell. That is the first universal common ancestor. Last universal common ancestor, particularly the archaea, bacteria, and eukarya, they are known as LUCA, or we can say it as universal common ancestor. Okay. Last universal common ancestor from which all life on earth evolved. So it is hypothesized to have existed around 3.5 to 4 billion years ago. We all know that from very close to level that life arise or origin of life on earth before 3.5 to 4 billion years ago. That's why it is also hypothesized that the universal common ancestors existed before that time. Okay. Another one important term is here, tree of life concept. That means the diagram represent the evolutionary relationship among all living organisms. Just look at this diagram. This is Luca. Here, my cursor is moving. This is Luca. Means last universal common ancestor. This is the ancestor from which all the life form they arise. And this is looking like a tree. That's why this is known as tree of life. Tree of life means its branches are actually showing which organism arise or what is the ancestral organism of other. So here Fuca is given first universal common ancestor. Fuca give rise to Leuca and ancient cells. But due course of time, this ancient cell, due to simplicity, they unable to adapt themselves with changing environment. That's why they move towards the extinction. So this tree showed their extinction. The ancient cell extinct due to their simplicity. But the last universal common ancestor, this is the last universal common ancestor, the bacteria, archaea, and eukarya. They develop their complexity, they diversify different groups, they form different organisms or different group of organisms, and those organisms survive. And evolutionary changes happen to those organisms, diversified group of organisms are formed, hence they survive in different forms. Okay, but ancient cell unable to survive. So this is the pictorial presentation. Then molecular evidence, the tree of life is largely supported by molecular data, especially the comparison of ribosomal RNA sequence, which shows deep evolutionary relationship. Okay, evolutionary relationship means the tree of life shows that eukaryotes, bacteria, and archaea have diverged from one common ancestor and connected through ancient evolutionary pathways. Just look at this picture. If we are talking about this group of organisms, just look at this blue pictures, the blue writings. Blue one, they the organisms developed from the bacterial group. That means primitive form was the bacterial form. Green non-sulfur bacteria, gram-positive bacteria, purple bacteria, cyanobacteria, lactobacteria, all this kind of bacteria they developed from this bacterial group in due course of time. If we are talking about archaea, then methano sarcina, methanobacterium, methanococcus, thermococcus, thermoproteus, pyrodictium, all these they form from the common ancestor. That is the evidence of life. That means life originated, a different form of life originated from one single ancestor. This is the tree of life. Okay, this is different kingdoms present under these three domains: the bacteria, archaea, and eukarya. Okay, so this is the introductory idea about three domains. And from this domain, different form of life. Here, just look at the picture. The eukarya give rise to animals, ciliates, green plants, fungi, flagellates, microsporidia. All these forms they are developed from this eukarya. So primarily these three forms were found, but primarily all these diversified groups were not found. But after that time, due to change in environment, due to change in environmental condition, these groups were formed. Okay, so this is basic idea regarding tree of life. This three domain system, bacteria, archaea, and eukarya, proposed by Carl Woese in 1990, which is based on differences in ribosomal RNA sequence. We know that different types of ribosome, particularly different type of RNA, rRNA, mRNA, tRNA, snRNA, different type of ribosomal RNAs, different type of RNAs group are there. But particularly for evolutionary point of view, the rRNA, ribosomal RNA, signifies its evolutionary evidences or provide the evolutionary evidences, particularly rRNA group. So this system divide life into three broad domains. So many micro domains are there, so many micro divisions are there, but entire division, entire broad division provided by these three domains, the idea provided by Carl Woese in 1990. Okay, just look at here certain examples in pictorial format. Actually, the idea provided by this 1977, but after that, this 1990, he proposed actual classification. So this archaea, bacteria, and eukarya, these are certain examples of this. Now we'll go for the characteristic. How? Basic, the domains are characterized, or what is the basic differences between the domains. Particularly if we discuss about the bacteria, they are prokaryotic group, otherly known as eubacteria, prokaryotic microorganisms without membrane bound organelles. That means nucleus, mitochondria, endoplasmic reticulum, whatever membrane bound organelles you can ask, they all lack in that group. But they are not similar with animal cell because they have cell walls, which is made up of peptidoglycan. Circular DNA, no histone associated with DNA. DNA is circular and no histone associated. DNA presents at the place of nucleoid because in eukaryotic cell, DNA is present in the nucleus. But here, no nucleus was found. Primitive nucleus was found, which is called as nucleoid. In that nucleoid position, DNA were present. Okay. They reproduce asexually by binary fission. some conditions, sexual reproductions are found. We'll discuss about it in later version, in later slides. They include diverse group such as cyanobacteria, proteobacteria, and all other bacteria were written in the previous slide. We have just discussed about that. Then, if we go for archaea or archaeobacteria, this particular bacteria domain known as eubacteria. Here, archaea group, otherly known as archaeobacteria. Archaeobacteria also prokaryotic group, but genetically distinct from bacteria. That is not same as bacteria. Cell membrane contain ether-like lipid, either linked lipid, ester link in bacteria and eukaryotes. Okay. Cell wall, like bacteria, they do not have peptidoglycan. Some have pseudopeptidoglycan structure. They resemble the structure of peptidoglycan, but they do not have actually peptidoglycan. Extreme mophiles, that means they thrive in extreme condition. They can adapt extreme condition like high temperature, acidity. Particularly why they adapt this extreme condition, because when they form, they actually face the extreme condition. That means at that time environment was extreme, environment was not so suitable. Okay. And more closely related to eukaryotes in terms of gene for replication. That means bacteria, if we compare bacteria with eukarya, bacteria with archaea and eukarya with archaea, then we'll get more similarities between archaea and eukarya in comparison to bacteria. That is the concluding mark. If we go for eukarya, the eukaryotic organisms with a true nucleus and membrane-bound organelles. These includes animals, plants, fungi, protists, linear chromosomes with histone protein. All these are the eukaryotic property, which is significant from the prokaryotic property. They have membrane-bound organelles. They have a double-stranded DNA with histone protein. They reproduce both sexually as well as asexually. Greater structural complexity than prokaryotes are found. So these are different basic characteristic division of these three domains. These are structural presentation of three domains: bacterial group, bacterial domain, archaeal domain, and eukarya domain. Okay. Now, what is the distinction between the domains? On which manner we can distinguish the domains? First of all, we'll discuss about genetic differences. Genetic differences, that means the primary division between these domains come from molecular differences. Okay. Molecular differences means the structure of ribosomal RNA, structure of DNA, their different genetic codes, replication mechanism, how RNA and DNA replicate, protein synthesis machinery, how RNA form the protein. All these kind of genetic differences we can found between the domains. Then not only the genetic differences, metabolic differences are also there. If organisms vary from one to another on genetic basis, metabolism is also affected by that genetic basis. That's why metabolism varies between domain, with archaea often having unique pathways suited to extreme environmental condition. Metabolism is also affected by environmental condition. Though archaeal group faced extreme environmental condition, their metabolism also reshaped accordingly. And the cell structure, obviously, another important distinguishing point. Prokaryotes, bacteria and archaea lack a nucleus and other membrane-bound organelle, while eukaryotes possess complex cellular structure. Okay. So these are basic differences between this group of organisms. Then the importance. Three domain system. What is the importance of this three domain system? Three domain system provides insight into the evolutionary history of life on Earth. What was the condition? How the life originated? What is the ancestor? Everything we can get idea from three domain system. Okay. Here at right side you can find the marks. Okay. Bacteria, archaea, they are simplest group of organisms. That's why so many divisions are not found. But in eukarya, you get different kinds of division due to their complexity. They are divided into kingdom, phylum, class, order, family, genus, species. All these are the different kinds of divisions we can find in case of bacteria. Sorry, in case of domain system. Evolutionary biology express the three domain system provide insight into evolutionary history of life. But if we'll discuss about systematics and taxonomy, it reshaped biological classification, challenging the previous two kingdom and five kingdom system by introducing domains that represent fundamental evolutionary lines. Okay, just look at here. Fundamental evolutionary lines means which organ give rise the other, or which organ is the ancestor of other organism. Okay, not organ, that is organism. Modern research say what? The system has been foundational in studying the origin of life, microbial diversity, and evolutionary process that differentiate archaea, bacteria, and eukaryote. Okay, so these systems shortly we will discuss in these classes. This is evolutionary significance of archaea and eukarya. Evolutionary significance means these two groups, they resemble more similar properties. Archaea and eukarya share more similarities in their genetic and cellular process compared to bacteria, suggesting a closer evolutionary relations. Okay, the evolutionary split between these domains represent a major transition in the history of life, with archaea likely giving rise to eukaryotic cell through symbiotic events. Just look at this picture at right side. How these organisms, bacteria position is here, but the position of eukaryotes and archaea are nearer to each other. It shows what? It shows when archaea give rise to other organisms, they first turn to this eukaryotic group, eukarya. Okay. In two domain system means archaea and bacteria were there. After that, eukarya formed from this archaeal group. Here also you can find in three domain system of life provided by Carl Uch. He also emphasized that archaea give rise to this eukarya. So this eukarya and archaea, from this structure we can conclude that these structural analysis showed that archaea and eukarya, they developed from a common ancestor. That's why they have more evolutionary significance. They have more similarities. They have more evolutionary significance. Okay, so this is the basic idea. And
Sir,
why they have
hello.
more evolutionary significance?
Sir,
Just little bit idea I will provide here. Detailed
sir,
discussion in next class will do. That is endosymbiotic theory. That means certain archaeal group, they engulf other bacteria or small cellular parts and they turn to eukarya. That's why archaea and eukarya, they share same or similar genomic sequences. Okay, just look at the picture in next slide. This is endosymbiotic theory. Endosymbiotic theory explains actually the origin of eukaryotes. Okay, how mitochondria and chloroplast established inside the eukaryotes. That is the origin of eukaryotes. Okay, just look at this picture. When photosynthesis was completely absent in the environmental condition, the cells generally looked for the food. It was hypothesized that the larger, bigger cells, they engulf certain smaller bacterial cells. The bigger cells were hypothesized as they are the archaeal cell. The smaller cells were the bacterial cell. When bacterial cell ingest into, this is the process by which endosymbiotic process by which the bacterial cell engulfed into the archaeal cell, and this bacteria make an symbiosis relationship with the archaeal cell because archaeal cell need energy. This bacterial cell have property to form the energy and store the energy. In that manner, scientists assume that these bacterial cell in latter part they converted to mitochondria. It was the assumption because inside the cell, mitochondria is the only organ which has its own DNA. It means all the properties of cell are maintained by cellular DNA which is present in nucleus, but only the bacterial cell, only the mitochondria in the mitochondria organ in the cell, they have their own DNA, mitochondrial DNA, which is known as the powerhouse. They help in synthesizing ATP. Okay, the substance, the powerhouse of the cell, because they break down fuel molecules, which is the capacity of another cell. They capture energy, they make adenosine triphosphate ATP, and the cell's mainly energy carrying molecule. That's why it was assumed that eukarya form in this pattern. Okay, that's why archaeal group had more relationship with the eukaryote group. Okay, and like that kind of theory for the chloroplast formation, but formation of chloroplasts, how chloroplasts are present in the cell, how they are incorporated in the cell, how mitochondria incorporated in the cell. That is the discussion part of next unit. For your knowledge, for your understanding, and just giving the idea how this endosymbiotic theory explained about the origin of eukarya, and the origin of eukarya explains the domain of life or significance between eukarya as well as archaea. Okay, so this is the condition. Am I audible, students?
Yes, sir.
Yes, sir. Yes, sir.
So this all about the first part, the introductory part about three domain system. I'm just sharing the second slide. Just wait for 1 to 2 minutes. Now I'll go for molecular phylogeny and molecular clock. So first we'll discuss about molecular phylogeny. What it refers is the study of evolutionary relationship among organisms using molecular sequence such as DNA, RNA, or protein. So, particularly, when we'll physically visualize the organism, it is very difficult to understand what is the actual ancestor of which organism. But the molecular data can provide a better idea about their ancestry, about their evolution. That's why the molecular tree was formed, from which common branch other sub branches are evolved. The evolution or the formation of these sub branches is due to this phylogeny analysis, or we can use the molecular sequence such as DNA, RNA, protein for this part. The method was developed a more objective approach to constructing phylogenetic tree compared to traditional morphological method, which can sometimes be misleading due to convergent evolution. If we'll discuss about convergent and divergent evolution means same organs having different function or origin is same, but due to diversified medium, due to diversified habitat selection, organisms show different function. But convergent evolution show what? Different origins are there, but the organs show similar function. Just look at these two pictures. For divergent evolution, if we look at the forelimb of birds, they meant for what? They meant for flying. The wings, actually the forelimbs are modified into wings, they meant for flying. The picture is not here, just for example. You all know that the four limbs of birds, they are modified to wings, and the wings are actually meant for flying. But same four limbs of whale, which is present in aquatic medium, modified to flippers, and these flippers are meant for swimming. So that condition is divergent evolution, because the two organisms having the same ancestry, because the four limbs shows the same ancestry, they all made up of humerus, radioulna, carpals, metacarpals, phalanges. This four limb structure means same in all the organisms what I have cited. But here, the organisms actually perform different function, because organisms, they share different habitat. Though their origin is same, but they show diversified group of organism due to diversified habitat. But why the convergent evolution example is given here? The ancestry, common ancestry condition is not difficult to understand in case of divergent evolution, because structural similarities are there. But in convergent evolution, if we'll go for the organismal variation, just look at here, the bird, the butterfly, the bat, they all have wings. But butterfly wings, they do not have any bones. Bird wings, they have certain bones, they have feathers. Bat wing structure is somehow different because this is mammalian group, this is insect group, and this is avian group. So ancestors are different, but they show similar function. If we place all the organism in evolutionary tree in similar group due to their function, then it would be difficult. That's why molecular phylogeny gives a complete different idea regarding this ancestry, where convergent evolution mislead the process of understanding the ancestry. Molecular phylogeny actually provide a better idea regarding this common ancestry. Okay, so use of molecular data for phylogeny began in the mid 20th century with early attempts relying on protein sequences. That means more advanced forms are there by which we can distinguish the organism, we can get a better idea about their ancestry, about the organismal ancestry. Okay. So this is all about different definitions and their history. Some key terms we should know before forming the molecular tree. The first one is phylogeny. Phylogeny is the evolutionary history of a species or group of related species. Molecular clock is a technique that uses mutation rate. That means in different generation, what was the mutation rate? So comparison between this mutation rate or comparison between mutation rates are expressed by this molecular clock. Homology is the similarity in DNA sequence. Here just look at the, if we are talking about globin gene, all these animals they have globin gene. Globin gene has two chain, alpha chain and beta chain. This is due to duplication, and in different organism, frog, human, mouse, they have alpha chain. That's why alpha chain is coming under the orthologous. That means orthologs are genes in different species that evolved from a common ancestral groups. Just look at here, the orthologs are the genes. That means the alpha genes. They develop from a common ancestor. That's why they are orthologs. But paralogs is the gene related by duplication within a genome. That means same mouse, they have alpha gene, they have beta gene. That is the paralog condition. Human also, we can compare this human alpha with human beta. They are also paralogs. Okay. So if we are talking about within organism or within the same group, within different organisms, we can assume these words, this homology. All these are homologous group because all they have globin genes. But homology is expressed in orthologous condition and paralogous condition. Orthologous means same organism, a different organism having same group of gene. Paralogous means within the organism, different genes are present due to duplication and divergence. Okay, so these are certain key terms we should know before constructing a phylogenetic tree. Many limitations are there for phylogenetic tree. Yes, it's no doubt the phylogenetic tree provide a lot of idea, help a lot to the people regarding different identification and ancestral evolution or evolutionary process. But some limitations are also there. What are those limitations? Limitation means on these factors, they unable to satisfy the reader or the scientist like that. Incomplete data. Sequencing may not cover the entire genome. That means, just take one example. When we are sampling, it doesn't mean that we can sample the all the species present in that area. Like that manner, when we are talking about sequencing, the genome, entire genome sequencing may not be possible in some cases, in some organism, which leading to incomplete data. That means we can provide the data, what information we have, but we cannot provide the data of all the gene present, all the information provided. Okay, that is the first limitation. Second limitation is the horizontal gene transfer. Particularly in bacteria, it is found horizontal gene transfer. Horizontal gene transfer, basically, we'll discuss about it in later slide. But this is the idea when gene is transferred from one species to another in horizontal level, but not from parent to offspring. In higher organism, vertical transmission occurs from parent to offspring, but in bacteria, that is not found from mother to daughter. Horizontal gene transfer occurs. Convergence is another limitation. Before sometimes we have discussed that convergent evolution mislead somewhere due to their similarities in function, but structural dissimilarities and computational complexity. When so many molecular data available to us, it is difficult for us to calculate all the data without advanced bioinformatics tools. Bioinformatics tools are different codes, different apps we have to use, we have to calculate the data because so many genetic codes are there. Okay. So this is the process by which we can construct a phylogenetic tree. This is the structure of phylogenetic tree. Okay, one by one, we'll discuss about it. So first thing, phylogenetic tree illustrate evolutionary relationship among organisms based on genetic sequences, similarities, and differences. This is the picture. Which organism is similar with other organism? On which manner and how the organism is dissimilar? What are the differences? And who, on based on which property they are different from each other? Everything we can understand from this phylogenetic tree. So what are the basic steps for formation of phylogenetic tree? The basic steps include three things. First one is data collection. Second one is sequence alignment. Third one is model selection. Okay, data collection, that means if we are talking about any organism, we have to collect the data first. Just look at this picture. Data means DNA, RNA, any kind of data. Data collection is the first. Then for different species, we can make table. For DNA, for the RNA, so many codes are there. And we have to mention these codes in this format, in this table. Then sequence alignment is very important. That means identify homologous region that can be compared. Just look at here. This is the picture where we get homologous region. Okay, this colorful region, similar color shows the homologous region in different sectors. Just look at here. This is different species presentation. Species A, that means this is different species. This is different. This is different. This is different. Outgroup means any other species. So for all the code 1 is the similar code. We can arrange them in one. We can color one. Okay. So where we get other similarities that we have to analyze after presentation. So for this presentation, collection of data is very important. And at last, model selection is very another important part, which were provided by Moto Kimura and Cantor. to scientists. They provide model selection. Choose an appropriate evolutionary model that describe how sequence change over time. Okay, that means that is the phylogenetic analysis. Just for this picture, for all the similar group, we can make one ancestry, but where it is different, we have to make certain branches. That means the difference point shows the differences or the branches. That means the organism at that level is differentiated from the previous, from the ancestor. By this, we can form here different tree, or we can construct tree like this. Okay, this is the basic presentation, basic idea regarding this. Okay, then tree building algorithm depends on what? Distance based method, neighbor joining, optima methods used. These are certain software methods. Okay, they are helpful for tree building. Character based method, maximum likelihood, and Bayesian interference are more sophisticated method using. That means we have to compare those organisms, those codes which have similarities or at which point they got difference. That is very important. This is the picture of the data. So the particular molecular database is an additional information that can be used to better understand this. It is a plant species presentation. The left hand side presentation is the evolutionary order of four species, four plant species. Actually, this is a picture from one research paper. Okay, so different group of species were aligned in this manner. Then this tree, the phylogenetic tree, was formed. Okay, here one presentation of animal is there, on which basis so many differences are there. It is mentioned here. Some organisms, their differences due to their feather. Some organisms, their differences due to presence of gizzard. Some lungs, they got differentiated. Okay, jaws, from this lamprey to this alligator, so many differences are there, and these differences are responsible for different branching. At last, we can form a tree. The branching shows its diversification from common ancestry. That means here in this picture, we can see that the common ancestry for all these animals are same. But from this common ancestry, certain organisms, particularly the lamprey-like organisms, are the aquatic organism, but aquatic organisms are differentiated by the presence of jaws. If we are talking about sea bass, they have jaws. That's why being aquatic, they are differentiated in this manner. Okay, so like this, so many other examples we can cite, so many other examples we can cite from different sources in our copy. Okay, so this is the basic idea how we form the phylogenetic tree. First, we have to collect the species. If we are talking about these species, we have to collect these species. We have to collect certain samples from these species. Then we'll analyze the data. How we'll analyze the data? Particularly some molecular data we have to collect. Molecular data means we have to isolate their DNA, isolate their RNA, protein, all these things. Then we'll segregate this. What is the code for RNA? What is the code, genetic code? Okay, so these codes we have to arrange in tabular manner like this. Then by arranging them, or which are the homologous group, we have to segregate, and by that we can form a tree. This is the basic idea regarding this phylogenetic tree. So tree for different three domains of life, from different domains, how organisms are diversified. This is a pictorial presentation. This is the basic idea. The root means here the common ancestry. Internal node, like a tree, the root means the basic common ancestry. Internal node means just this common ancestor divided into two. So many branches, that means they have certain other characteristics which are not present in the previous groups. That's why they branched into diversified group of organisms like this. Okay. So this is all about phylogenetic tree and its construction. Now, we can also make phylogenetic tree based on 16S rRNA. Ribosomal RNA, before some time, I have told you that ribosomal RNA played an important role in evolutionary expression. Where we get this 16S rRNA? When we are talking about prokaryotic ribosome or eukaryotic ribosome, we have certain large unit, large subunit as well as small subunit. Okay. So here the pictorial presentation of prokaryotic cell is there. Prokaryotic cell, small subunit contain this 16S ribosomal RNA because this RNA is present associated with this ribosome. That's why that is ribosomal RNA. And this ribosomal RNA, again, if we fragment this 16S according to its bedworks unit, it is divided into 16S rRNA. Okay, this 16S rRNA is actually providing idea. These are the different segments. Okay, variable region, conserved region. So by assuming, by comparing this region, we can compare the organismal group. Different organismal 16S RNA can provide one idea about the organismal ancestry. Okay, this is the basic method by which we can collect the data. We have to collect the sample from the species. Then these samples are subjected to extraction. That means DNA, RNA, protein extraction by microcentrifuge. After extraction, PCR polymerase chain reaction should be done for the analysis. Then after analysis, sequencing of DNA. That means we have to arrange the pattern. Then we have to form a library. Library means the tabular presentation of the data, and then we can compare the data. Okay, so this is the data analysis by using 16S rRNA gene. Okay, these are the procedures already mentioned here. PCR amplification, that is the extract bacterial DNA and amplify the 16S rRNA gene using PCR. Sequencing, sequence the PCR product. Sequencing alignment is the align the sequences from different bacteria. This is the aligning, sequencing. Okay, that means we have to arrange. Then reconstruction by making the library. Library construction is actually the backbone for the tree construction. Okay, and at last tree interpretation, that means it helps in for evolutionary relationship. When the complete tree is formed, that actually interpret the evolutionary relationship. Okay, this is the idea. This is the phylogenetic tree formation using 16S rRNA. Then for this part, this is another important concept, that is concept of speciation in bacteria. That means how different species of bacteria are actually formed. Okay, so this is the different constructs. Speciation. Speciation means formation of new species. How different species are formed. It is more complex than multicellular organism. because horizontal gene transfer is there. Horizontal gene transfer means here you can see all the genes are transferred from side to side manner, not from parent to offspring manner. Nowhere you can find one is parent, another is offspring. One bacteria give the things to another bacteria. They cannot produce a new bacteria by their conjugation, by any mode of reproduction. But they can share certain parts of their body to other organisms. That's why that is horizontal gene transfer manner. Recombination and OTU, operational taxonomic unit. That means sequencing data using 16S rRNA gene similarity threshold. Okay, so these are actually responsible for this bacterial speciation, and this bacterial speciation actually we'll discuss in the last slide because it has a great importance. Bacterial speciation. Ecological speciation, bacteria can diverse into distinct species through adaptation to different ecological niche. Okay, that means when bacteria are subjected to different ecological conditions, they got actually different challenges, and to mitigate, to adapt themselves in that changing environmental condition, they accept those challenges and they modify themselves to develop their resistance in accordance to their challenges. That's why they actually modified their genetic material. If one genetic material is different from other genetic material, then we can say the new organism is formed. That's why these 3 points are very important. Conjugation, transformation, and transduction is the actual pathway for this horizontal gene transfer, and by this they share their gene, the genetic material, with the other bacterial species. Okay, and the biological species concept based on reproductive isolation is difficult to apply to bacteria. So species are often defined based on genetic divergence and ecological factor. They are small organisms. We cannot separate them by biological speciation group, reproductive isolation group. Okay, so it is best to separate them by horizontal gene transfer or by their analysis of genetic material. Now, the next important concept we'll discuss what is called as molecular clock, molecular divergence and molecular clock. So the previous one is one part of the second unit. It is another part of the second unit, the description about the molecular clocks. How molecular clock is formed, what is the importance of molecular clock, molecular divergence, how helps in forming the molecular clock. So first we should know about the molecular divergence. What it refers actually, molecular divergence refers to the process by which genetic sequences in different species accumulate mutation over time. That means if we are discussing about the phylogenetic tree, here just look at this picture. One group of organism, the common group of organism, to diversification is there means at this certain point the common group adapts to new environment, or mutation occurs at this point at a particular gene or a particular level which lead these organisms into 2 different groups. Okay, again this organism has its own individual property. This organism more similarity with the parent organism. But this diversification is completely different from this parent diversification. Okay. So here we can assume, we can compare how the mutation occur and on which point the mutation occur. Like that, in all the points of diversification, we find that the mutation, and we can compare those mutations. So presence of this comparison is known as molecular clock, basically. This is due to genetic variance or molecular divergence. It refers the process by which genetic sequence in different species accumulate mutation over time. This is one species, this is another species. How they are different? They have common ancestry, but they differentiate from each other by accumulation. That means generation after generation, they accumulate certain kind of mutation at a particular level of gene or their genetic property. If mutation is accumulated generation after generation, that leads to what? That leads to change in species characteristic, which leads to formation of new species, speciation. Okay, this is the diagrammatic presentation of human evolution, formation of human tree. Okay, evolutionary tree. Now, come to the concept of molecular clock, which was provided by Emile Zucker-Candle. Okay, and another scientist also have important role known as Pauling in 1960s. What it provide the idea, actually, the genetic mutations accumulate at a relative constant rate over time for certain gene of protein or proteins. That means, particularly if the gene A or B, we can take any kind of like example. If a gene is affected by certain environmental factor in every generation, that means, and the change is suitable for the organism. That means organism in every generation accumulate certain amount of mutation. If you look at this picture here, from this common ancestry, this group is different from this group because this group having the property, but this group accumulate mutation after mutation at certain point, and at last they form a new species. That means the mutation from this and the mutation from this, generation after generation, the chimpanzees accumulate, certain group of chimpanzees, they accumulate certain mutation generation after generation, they form Neanderthals. And these Neanderthals, they accumulate mutation generation after generation, they form the modern human being. This is the case. Okay, we can compare these mutations and express it. The format is known as molecular clock. Okay, at a particular fixed rate, the mutation is accumulated generation after generation. So by comparing the molecular differences between species and using known rate of mutation, that means the molecular clock, scientists can estimate when species diverged from a common ancestor. Okay, so after tree formation, if we add the time, how and when, then only we can express the data of molecular clock. Clock calibration means the measure must clock must be calibrated using fossil records. That means the comparison is yes or no, true or false. That can be calibrated by other historical data, whatever written in different format, the history. Okay, fossil records are there. whether our evaluation is correct or not, we can compare those evaluation with fossil records. If fossil records and our evaluation are almost similar, then our evaluation is correct. Okay, this is the proof for the fossil record, the present proof for the fossil record. That is the importance of molecular clock. Just look at this picture. Distance-based maximum likelihood, the Bayesian method, the genetic data collection was there, DNA sequences, protein sequences. Then calibration is essential. Calibration means checking by different fossil record. Okay, so combined form model selection, clock model, presentation of this, variation of this comparison, and molecular dating analysis provide the idea. Molecular dating analysis means we can assume the time by carbon dating, uranium dating, different dating processes are there. Okay, then only we can interpret which one actually the ancestor of other. Okay, these are the pictorial presentation of human molecular clock, how one organism is diversified from another organism, and what is the time period, million years ago time period, the organism. Here not million year ago, it is written in year format. Okay, and how the organism is differentiated from one another with the presentation data. And the first picture, molecular clock, is shown here. From common ancestor, certain genes were analyzed part, where the mutations occur, where first mutation occur, where second mutation occur, how we compare those mutation in modern sequences, everything we can get idea about molecular clock. It can give us idea about the mutation rate. That means here, particularly in this picture, one mutation every 30 million years. That means in yesterday's class, we have discussed that evolution is a completely slow process. It is not fast process. Generation after generation, thousands, lakhs of generation required for a single change. Okay, here you can see that one mutation is fixed for that species. That means change in species in 30 million years. Particularly a single characteristic change, it takes 30 million years. Okay, so this is the clock which provide the idea about the mutation rate, the form of evolution. We have discussed that mutation is the backbone for evolution. If we can calculate the mutation in different stage, we can calculate the mutation, rate of mutation in different generations, we can assume the process of evolution. We can assume the ancestry of the species present. Okay, so this is all about molecular clock. The mechanism of molecular clock explains what? The rate of mutation in a particular gene or genome can be thought of as a tick of the molecular clock. Okay, that means the mutation actually shows the time where mutation occur. These are different genes, and different genes shows the rate of mutation. Where the mutation occur can be assumed by observing different gene and their position. Okay, which gene is actually responsible for which factor? If the factor, the process, if the particular gene is responsible for metabolism, then where we got the information about change in metabolism, that means there is a mutation in that particular gene. Okay, Some genes evolve faster than others with non-coding regions or synonymous substitution, which do not affect protein function, often evolving faster than coding regions. Okay, that means there is different type of mutations. Faster mutations are there, slower mutations are there, but if the mutation is constant, that may take longer period of time, that leads to the process of evolution, and that data is only provided by using this molecular clock. Okay, for this molecular clock, one neutral theory was provided by Moto Kimura. Moto Kimura contributed a lot for this molecular clock analysis, and his theory on this molecular clock supports the idea that most evolutionary changes at the molecular level are neutral. That means they do not affect an organism's fitness and accumulate at a relatively constant rate, providing the foundation for molecular clock. That means more mutations are constant. It is actually provide the idea about, or it helps us in comparing the mutation, where the mutation actually happens, where the change actually happens in different generations. We can get proper idea about that. That is the neutral theory of molecular evolution. Then we'll go for molecular drive, another important concept. What actually the molecular drive is? It is the process by which genetic changes spread through a population, not necessarily by natural selection, but by mechanism such as gene conversion, biased gene conversion, unequal crossing over. That means in previous class we discussed about natural selection. Natural selection explains about the fitness. That means certain changes happen in the species in comparison to other species. But what is the reason for that fitness, for that change? The particular reason is, just look at this picture at the right side. When sexual reproduction is there, just look at these two genome present in different gametes. Okay, this is the structure, this is another structure. How this molecular drive, that means the process by which genetic changes spread through a population. That means if migration is there, in previous class, just remind migration, gene flow, gene migration that leads to evolution. If this is the male sperm from another population come to the particular area population where the female and its particular male has different genetic structure. But when this completely new gene is combined with this old or that particular area gene, the offspring they form having different genetic structure like this. The offspring may be male or female. Okay. But the offspring, they have completely different kind of gene from the parents, some of the offsprings. That leads to the process of evolution, which is expressed by molecular drive, because molecular drive emphasized on genetic changes spread through a population. Okay. So when the population having changed version of its gene, which is very essential for the population. If a population is particularly affected by any kind of disease, if a particular gene is responsible for the death of the population, then change in gene is very important. That is expressed by this molecular drive. How this change occur? When another gene or another property of another gene is incorporated to that gene, and that is possible due to this crossing over process. Just look at this picture. Here wild type genes are there, okay, and some drive parents means variation genes are there. When this wild type is crossed over this variation, that means certain portions are exchanged by each other. Just look at this picture. This green color is incorporated here in the new gene. The green color is incorporated in this, and some of these colorful segments of the gene drive parent are incorporated into the wild parent. Okay, so this is the change actually happened due to the crossing over process. And this crossing over is actually responsible for this kind of changes during the process of sexual reproduction. So molecular drive can lead to the rapid spread of certain genetic changes within population, sometimes complicating the interpretation of evolutionary histories, especially when different mechanisms of molecular drive occur at different rates. Molecular drive explains what? It explains about variations. If variations are there, then new descendants can be formed, which are genetically and reproductively isolated from previous organism. If new descendants are formed, the new descendants lead to formation of new branch in the tree. Okay, that means that sub branch are formed, new organisms are formed. Okay, so this is the case of molecular drive. So molecular clock and molecular drive both are important. Molecular clock explains us what? Molecular clock explains us about the rate of mutation. For ancestral derivation, rate of mutation is very important. Phylogenetic tree explain the idea about the ancestry, but molecular clock adds something on this phylogenetic tree, because molecular clock provide the idea about time, before which how many times they are formed, or million years ago, billion years ago, how the organism, what are the organisms, what are the ancestral organisms were present. Okay, different for this process, different dating processes are also there. I have already told you carbon dating, uranium dating. This also provides certain aid to analyzing this data. Okay, so these all are about molecular phylogeny, the tree construction, phylogenetic tree construction, molecular clock, and molecular drive. Okay, now we'll go for the last part of this unit, that is bacterial speciation. How speciation occurs in bacteria. For that, I'm just sharing a new slide to you. Speciation in bacteria, or we can say it as bacterial genome evolution. How bacterial genome or evolution of bacterial genome occurs. Okay, so for this, horizontal gene transfer is playing an important role. What it actually explains, horizontal gene transfer actually explain about the process by which bacteria acquire genetic material from other organisms rather than inheriting it from parent organism. Here, we can distinguish between vertical gene transfer and horizontal gene transfer. Just look at these two words written here, VGT and SGT. They are differentiated, they are different from each other on this manner that if the organism acquiring genetic material from other organism, from other sources, apart from their parents, then that kind of gene transfer is known as horizontal gene transfer. Horizontal gene transfer. But on the other hand, if the organism acquiring gene from its parent, that is known as vertical gene transfer. What happens in our case? We have all the genes which were acquired from our parent. We cannot receive the gene from other sources. We only can receive the gene from our parents. That's right. Horizontal gene transfer is not found in our case. We can express the vertical gene transfer method. Horizontal gene transfer is generally found in case of bacteria. That mechanism played a significant role in evolution of bacteria because so many other organisms, so many other factors are responsible for bacterial evolution. Because if gene transfer within the same species occurs, that do not lead to evolution of the species or immediate evolution of the species or change in species in a very fast manner. Though the bacteria, they receive the gene from other sources. That means other sources, variations influence the bacterial gene, which leads to the evolution. Okay. And this horizontal gene transfer allows for the acquisition of genes that provide advantages such as antibiotic resistance, virulence factors, and metabolic capabilities. Okay, so these are different factors by which bacteria benefited from this horizontal gene transfer. Evolution occurs, but along with evolution, certain factors they're responsible for bacterial benefits. Like bacterial antibiotic resistance is possible due to this horizontal gene transfer. Okay, what is the basic mechanism for this horizontal gene transfer? Three basic mechanisms are there. One by one, we'll discuss about it. Transformation is one of the best mechanism, conjugation and transduction. So these are three different mechanisms for horizontal gene transfer. First of all, we'll discuss about this transformation. Transformation explains actually what it explains the process of DNA uptake by the bacteria from surrounding environment. The cells that have the ability to uptake DNA are known as competent cells, and this process was first reported in Streptococcus pneumoniae by Griffith. We all know that Griffith experiment. What Griffith experiment explains? Frederick Griffith, he explained about Streptococcus pneumoniae. And in his experiment, he actually explains a very interesting thing. So, what was the experiment provided by Griffith? He injected A strain and R strain to mice. Okay, just I'm explaining due to short span of time. I'm just explaining. No need to elaborate it in a very simple version, in a very quiz, just glance analysis. I'm just explaining the Griffith experiment. He injected smooth and rough strain to mice. Rough strain means the bacterial strain to the mice. The one which was injected with, that means the mice which was injected with the smooth strain, they developed pneumonia and died, while the infected with R strain stayed alive. That means the smooth strain is the dangerous strain, harmful strain, and the R strain is the harmless strain for the mice. But what he did in his second group of experiment, he just injected heat killed A strain. We know that A strain in its living condition is harmful for the mice. But when heat killed A strain was injected into mice, the mice stayed alive. That means he concluded that heat killed means they completely destroyed the genetic property of the bacteria. That's why the mouse was not affected by pneumonia and the mouse was alive. But when he mixed, that is the experiment of this bacterial transformation, what picture is showing here. When he mixed with heat kill strain, S strain, and R strain, in other condition, R strain is harmless. Heat killed strain also harmless. But when he mixed both, that means the R strain, the harmless strain, receive the A strain gene and get infectious. Okay, just look at this picture here. If this is the heat kill strain, the first blue picture is the heat kill strain, and this is the R strain. This R strain, the raw strain, which is harmless, they receive this heat killed form, and the heat kill gene is getting incorporated into the bacterial genome part. Plasmid is extra chromosomal DNA. Normally, chromosomal DNA are different, but plasmid is extra chromosomal DNA present in case of bacteria, and those plasmids they receive the outer DNA. That means the DNA has no relationship with this bacteria. That is completely environmental DNA, completely the outer DNA. What the definition shows to us? Transformation is the process of DNA uptake by bacteria from surrounding environment. So this is the surrounding environment. And from this surrounding environment, the bacteria receive the DNA. Okay. And when the enzyme DNA ligase cut the portion and incorporate the DNA into the bacterial DNA, they become the part of this bacterial DNA. And all the properties of the parent bacterial cell come to the new bacterial cell. That means the A strain property is coming to the R strain, which was primitively harmless, become harmful for the mice, and the mice become dead. That is the one process of horizontal gene transformation, and the horizontal gene transformation is actually responsible for bacterial evolution. Okay, so this is very interesting approach of Griffith experiment, and it explains about bacterial transformation. Then the second process, the conjugation. What conjugation actually explains? The direct transfer of DNA between bacteria through cell. cell to cell contact is a kind of reproduction which is considered as sexual reproduction in bacteria. Okay, so what happens? That is the peculiar type of reproduction in bacteria. What happens in conjugation? Direct transformation of DNA between bacteria through cell to cell contact, often mediated by plasmids. Just look at these pictures. Why we are calling as horizontal gene transfer? Because this is one bacteria, this is another bacteria. This bacteria having a peculiar property known as pilus, which is known as an outgrowth. It is sex pilus. And one plasmid is there. Plasmid is, this is one genome. The chromosomal DNA is present here. This is the nucleoid segment. But bacteria having the DNA other than this nucleoid, which is known as extra chromosomal DNA. Okay, and this extra chromosomal DNA is called as plasmid. This is one kind of bacteria. This is another kind of bacteria. There is no evolutionary relationship. These are completely different. But how this bacteria affect this bacteria and formation of new bacteria occurs? Formation of new bacteria means not parturition, not birth of new bacteria. The same bacteria change its property into another. That is the formation. That is the process of evolution in bacteria. So what happens here? Just look at this. The donors, that means who have more property. Here, the first one having more property, presence of this plasmid DNA. Okay, but the second one, they do not have this property. So the donor is considered as A plus. The recipient is considered as F minus. A plus donor, F minus recipient. The donor cell F plus from a sex pilus and begin contact with F minus having no sex pilus. Sex pilus is a tube-like structure which can be incorporated from one bacteria to another bacteria for transmission of this genetic material. Okay. So when there is physical contact due to any manner, if they are staying in aquatic medium due to any movement of water or any kind of physical contact happen between these two, the sex pilus is getting attached to next bacteria. That means the sex pilus of donor bacteria is getting attached to the sex pilus of, to the body of recipient bacteria. And this sex pilus make a connection between two bacteria, make a bridge between two bacteria through which they can interact with each other. And certain enzyme, relaxosome and transferosome, they play an important role in cutting this segment of extra chromosomal DNA of fast bacteria and transformation of this to the next bacteria. And after certain time, the donor is separated from the recipient by donating certain extra chromosomal material, and the cutting extra chromosomal material again replicated, and the bacteria as usual like previous form, but the new thing happens here. What is that? The bacteria, just look at this first picture, they didn't have sex pilus, they didn't have extra chromosomal DNA, but after this conjugation process they develop sex pilus as well as extra chromosomal DNA. Okay. So this is the case of bacterial conjugation. The donor and the recipient strand both contain a single strand of a plasmid. The complementary strand is synthesized in both the recipient and the donor. So the recipient cell now contain a copy of a plasmid and they become the new form. They can donate the same thing to other bacteria which do not have this property. So this is another horizontal gene transfer method because no organism receives its gene from the parent organism. This organism, they do not receive the gene from its own parent. They receive the gene from others. That's why that process of transformation is in horizontal manner, which is called as horizontal gene transfer. Another one process is there for this horizontal gene transfer that is known as bacterial transduction. That means bacteria receive the gene from the recipient. That means the recipient received the gene from the donor, not directly, by bacteriophage. That is very important. The virus, bacteriophage at the virus. What this process actually explains? Just 1 minute. This transduction process where virus known as bacteriophage transfers genetic material from one bacterium to another bacterium. Just look at here. This is the bacteriophage associated with the bacteria. Bacteriophage means the virus associated with the bacteria. Okay. Then this bacteriophage actually responsible for transformation of genetic material, genetic transfer from one bacteria to another bacteria. Okay. There is no direct contact of this bacteria. Just look at this picture. There is no direct contact of this bacteria. The bacteriophage which infect the bacteria, when bacteriophage is associated with the bacteria, they start infecting the bacteria. Okay. By using host, bacteriophage has its own DNA. This is bacterial DNA. This color is bacteriophage DNA. Okay. When bacteriophage incorporates its own DNA, own DNA or RNA, any kind of nucleic acid material into the bacterial cell, they start multiplying in the bacterial cell because bacterial cell is the host for virus. Virus is non-living outside the host, but it become very active when it come inside the host. That means particularly the nucleic acid substances material they develops, they get replicated, they change their number, they expand their number in a progressive manner inside the cell. Okay. Later, when this kind of virus, this is the multiplication of viral genome inside the bacterial cell, and this viral genome When aggregate certain amount of protein, they become this kind of viruses. That means these viruses having bacterial genome property as well as viral genome property. Okay. So when this kind of virus affect other bacteria, they transfer this bacterial property to the next bacteria along with the viral property. So transduction is commonly used in genetic engineering for inserting foreign DNA into the host cell. Okay, this is the benefit. But by this process, horizontal gene transfer occurs. This is not the parent of this bacteria, but this get the property of this bacteria due to this bacteriophage infection. Okay, this is all about bacterial transduction. So these are three elaborated form of horizontal gene transfer mechanism: transduction, transformation, as well as conjugation. This is the role of plasmid in DNA transfer. So particularly the plasmids, why they are important? They are small. This is the position of plasmid here. Just look at this. These are the plasmids. They are small circular extra chromosomal DNA molecules that replicate independently of the bacterial chromosome. That means their replication do not depend the bacterial chromosomal DNA replication. They independently replicate. Plasmids often carry genes that provide selective advantage to bacteria, such as antibiotic resistance, virulence factors, or metabolic genes. So they are important. Before sometimes we got to know that plasmids are very important in gene transfer. Okay. So here the antibiotic resistance plasmid example is here for you. Here the air plasmid, that means the plasmid contain the genetic material which develop antibiotic resistance in bacteria, which is considered as the donor. The recipient, they become dead because they do not have antibiotic resistance factor. When conjugation occurs between these two, that means the R plasmid having bacteria, when transferred to the R plasmid devoid bacteria through the sex pilus, that develops the antibiotic resistance nature by transconjugation process. And now the bacteria do not die due to presence of antibiotic. Okay, or that same dose of antibiotic when previously it was dead. That's why that is one of the example I have cited. So many examples are there why this plasmid. Nowadays, in a very case, we use those plasmid as vector for incorporating certain essential genetic information into the other cells. Okay. Here conjugative plasmids are there. Some plasmids like A plasmid in Escherichia coli. We have already discussed about A plasmid. They facilitate transfer of DNA between bacteria by conjugation. These plasmids encode the machinery needed to form a pilus and initiate DNA transfer. Non-conjugative plasmids, these plasmids rely on conjugative plasmids to be transferred but can still carry important genes. Okay, so these are basic processes we have discussed. By these processes, bacteria got their evolutionary significance. Just look at here. Already we have discussed in one slide If evolution, horizontal gene transfer, just look at this. Yes. Why this horizontal gene transfer is there? Horizontal gene transfer is there for their evolution. But in certain cases, horizontal gene transfer provide advantages. Advantages for what? Here particular example is cited, that is for antibiotic resistance. I have also given here antibiotic resistance factor. Why? Antibiotic resistance developed in one bacteria. What is the cause for antibiotic resistance? That means the bacteria do not have antibiotic resistance property. They develop antibiotic resistance property due to this evolutionary changes. Okay. And for that, horizontal gene transfer is playing an important role. Okay. This is the backbone for which there is speciation in bacteria occurs. Speciation is due to evolution in bacterial genome. Bacterial genome in different time evolve from one to other. Okay, that leads to their evolution. Now come to the last part of this unit, that is the transposons. Transposons are the jumping genes. The very interesting name for this transposon. They are actually the jumping genes. Okay. They are the DNA sequence that can move from one location in the genome to another location, either within the same DNA molecule or between different DNA molecule. Okay, that is the peculiar characteristics. So types of transposon we will discuss about: insertion sequence transposon, composite transposon. Transposon is actually you can say that transposition is the process in which bacteria involves the movement of genetic element, which are called as the transposon, the jumping genes, to different location in the genome. If a single location contains a particular fragment, on the next segment, on the next day, the other location contains the fragment. That means there is change. Transposons are responsible for that change that leads to the process of evolution. Okay, so this transposition can be replicative or non-replicative. That means in non-replicative transposition, the double helix is cut at the original site before transposon moves. In replicative transposition, the transposon is copied at the insertion site and the origin site without cutting the double helix. So what are the effects of this transposition? They can lead to duplication of genetic information, which can change genome size and modified gene expression pattern. If once genome size and gene expression patterns are changed, that leads to the change in genome. We can say it as the evolution of genome. Okay. So transposition mobility can be autonomous or non-autonomous. Autonomous transposition can move by themselves, while non-autonomous transposition require another transposition to move. So these are certain pictorial presentation for this jumping gene. Certain examples of transposition. Mu DNA transposition is an example of transposition that has been studied extensively because it can randomly insert into bacterial chromosome. Okay, so these are basic ideas regarding transposons and transposition. Transposition is the process by which transposon they change their location. If once the location is changed, then that leads to formation of new segment. Formation of new segment means there is complete change in the genetic makeup. Complete change in genetic makeup means whatever functional aspects, whatever functions performed by the old gene, it change is modified. That is the role of the transposons. Okay, so this all about different aspect of this unit. If anything left, anything not understood, then you people can ask me. So now, if any doubts, for 3 to 5 minutes, we can discuss about some doubts. You can ask me if time permit. I'll make clear your doubt in this stipulated time. Otherwise, we'll discuss it in later.
Good evening, sir.
Okay. Now you can ask me
Good
if
evening,
any
sir.
doubt.
Hello. Hello.
We have some times. We have 5 to 10
Sir,
minutes. We can discuss something. Yes,
sir, can you please explain the transpositions once again?
the transposons.
Yes, sir.
The transposons are actually in simple word. How we explain it? Just think about the DNA. Okay, DNA contains certain genes. Yes or no?
Yes, sir.
If the fragment, the genes of those DNA are not constant, they are changing their location. That means how they change their location? By normally, if we are talking about jumping manner, we can change our location. So that particular genes, not all the genes of DNA, that particular genes, those who change their location by jumping or particularly changing manner, they are called as transposons. Okay. In other words, we can say that how they are important. Transposons alter the genetic identity. That means if the gene location in the DNA segment is changing its position from one to another, the previous DNA, whatever function performed by the previous DNA, now the function is getting altered or not, changed or not. That is the importance of this transposition. That means transposition by the process of transposition can create or reverse the mutation. If the
Sir.
mutation occurs at any position, the mutation may be harmful, but due to transposition, mutation means complete stable change of this segment of gene. But due to transposition, maybe that gene come to its original position. That may reverse the original condition. Okay, that is the importance of transposition. It is also important because it duplicate genetic material. Duplication of genetic material happens due to this transposition. So basically, that Transposon is the jumping segment of DNA. Jumping segment means the gene which jump from one location to another location. That's why they are playing a very efficient role in bacterial evolution. They can transfer like antibiotic resistance gene between bacteria. They can promote chromosomal arrangement and more other functions they can perform. That's why they are very important. Okay. Now,
Yes, sir. Got it.
got some idea
Thank
about
you,
this
sir.
transposon?
Yes, sir.
Okay. Any
Thank you,
other
sir.
confusions? If any confusions, if any other confusions you have, then you can pin me through your WhatsApp. Okay. So
Okay, sir.
for today,
Yes, sir. Yes, sir.
we'll wind up here the meeting.
Yes, sir. Yes, sir. Thank you, sir.
Okay. Thanks to all for your patience and very good night to all.
Thank you,
Thank
sir.
you.
Good night.
Night, sir.